Hi there, everyone. Let's talk a little bit about blood typing. So, your blood type, I'm sure, are aware, has a lot to do with your eligibility to receive blood transfusions, donate blood for transfusions, organ transplants, and things of that nature. So, let's talk a little bit about what determines your blood type. So, your blood type has everything to do with your red blood cells. Your red blood cells, on the surface of their plasma membrane, have types of glycoproteins, which we are going to call antigens. Now, these are not the only types of antigens that are found in nature or even in your body. There are millions of different types of antigens. So an antigen is just a substance or a molecule that interacts with another type of substance called an antibody, and we'll have more to say about that later. But these antigens are just glycoproteins that are found on the surface of your red blood cells. And for our purposes, for most blood typing procedures, there are three different types of antigens. There is A antigens, B antigens, and then a third type, which is called the Rh factor, or Rh antigen. So your red blood cells can have these three antigens in any different combination possible. Your red blood cells could have all three of these antigens, none of these antigens, one of them, or two of them. They are all possible, and the different combinations that are possible is going to be what determines your blood type. So let's start with the simplest of the blood typing systems, which is called the ABO system, probably what you're most familiar with already. The ABO system has everything to do with these first two antigens, the A and the B antigens. So, for this system, we have four different possible blood types, and this, again, is determined by the types of antigens which are found on the surface of your red blood cells. So, in this first possibility, let's say that someone only has A antigens. So, the surface of the red blood cell is going to be studded with these A antigens, but not any B antigens. So in this case, if these are A antigens, this person would be blood type A. In another case, if someone, instead of having exclusively A antigens, instead had B antigens, as you might predict, this person would not be type A, but rather would be type B. So these first two possibilities describe scenarios in which you either only have A antigens or only have B antigens. Okay, well, what if you actually have both of them? So in this scenario, not only do we have A antigens, but we also have B antigens. So essentially you have both types of antigens. So in this case, again, as you might predict, if having just A antigens makes you type A, if having just B antigens makes you type B, having both of them would cause you to be type AB. So of the four combinations we're going to talk about here for the ABO system, we've covered having just A, just B, both A and B. So the only possible combination that is left would be, what if you have neither A nor B? And that is totally possible, and that is going to produce blood type O, in which you have neither A antigens nor B antigens. Okay, so now that we've talked about the ABO system, let's bring in the Rh factor. So the Rh factor is going to complete our discussion here. So the ABO system and the Rh system are not mutually exclusive. They actually coexist, and knowledge of both types of blood is going to be necessary for determining eligibility for transfusion procedures and other types of things. So the Rh factor, or Rh antigen, is just another type of antigen that exists on the surface of red blood cells also, but it is the only one of its kind, basically. There are several different kinds, but for our purposes, we're going to assume it's just one. So basically with the Rh factor, it's the same deal. You either have it or you don't. You either have it or you don't. So instead of modifying the ABO system, instead of saying you're either type A Rh or you're just type B, or you're type AB Rh or type O Rh, to indicate whether the Rh antigen is present or not, we modify the ABO blood type with either a positive or a negative. So if you are type A over here and you have the Rh antigen in addition to your A antigens, we would say that this person is type A positive. Type A positive, the A indicating that you have A antigens, the positive indicating that you have Rh antigens. If you are type B, but you do not have the Rh factor, we would say that you are type B negative. And then the same applies for A, B, and O as well. If you are type AB, but you do not have the Rh factor, we would say you are AB negative. And if you are type O, and the only antigen you have is the Rh factor, then we would say that you are O positive. So between the ABO system and the Rh system, we have basically 3 different types of antigens that can occur in a number of different combinations. And altogether, this is going to produce 8 different possible blood types: A positive and A negative, B positive and B negative, AB positive and AB negative, and O positive and O negative. So as we are going to see, knowing blood types for both a recipient and a donor, the donor being the person who is donating blood and the recipient obviously being the one receiving it, knowing the blood types of both people who are involved, both the donor and the recipient, is going to be essential to determining whether this transfusion can proceed as planned, whether there's going to be any problems or whether everything can go ahead as scheduled. But what is it about these blood types that makes certain blood types incompatible with others in the terms of transfusions? So to understand that, we really need to go beyond our discussion of antigens. Instead, we need to talk about a different type of molecule called antibodies. So when we defined an antigen before, we said that it is any type of molecule that is specifically recognized by a particular antibody. An antibody is a very complex protein structure that looks something like this. It has what is called a heavy chain. It's got 2 heavy chains actually that are connected together by disulfide bonds. And then it's got 2 light chains, which are attached to the heavy chain, again, by disulfide bond. It's basically this big Y-looking molecule. So here, on these 2 parts that I'm highlighting with these red circles, those are antigen binding sites. Every antibody is different. Each antibody has a particular type of antigen that it is going to go looking for. So, when you're talking about your blood type, for each blood type that we considered, that blood type is going to have a different type of antibody that is present in the blood plasma. So the antigens are present on the surface of the red blood cells, and the antibodies are floating around free in the blood plasma. So here's the idea. Whatever your blood type is, meaning whatever antigen you have, you are not going to have the antibody that recognizes the antigen that you have. But you are going to have the antibodies against the antigens you do not have. So let's see what we mean here. So for someone that is type A positive, meaning that they have A antigens and they have Rh antigens, they will not have anti-A or anti-Rh antibodies, but what they will have is anti-B antibodies. Someone who is type B negative, meaning that they only have B antigens but not A or Rh antigens, they will have anti-A and anti-Rh antigens floating around in their plasma. Someone who is type AB, in which they have both A and B antigens but no Rh antigens, they will have only anti-Rh antibodies. And then someone who is type O positive, in which they have the Rh antigen but no A or B, their plasma will contain both anti-A and anti-B antibodies. So understanding how your blood type not only determines what antigens you have, but what antibodies you have floating around in your blood plasma is essential to understanding and being able to predict when a blood transfusion can occur and when it should be avoided. But what is it about these antigens and these antibodies that makes such a thing so important for us to be on the lookout for? So the concern is when an antigen on a red blood cell is bound to and recognized by an antibody. So let's take a look at a red blood cell here. Let's say that this is a type A red blood cell. So it's got its A antigens littered all over the place here. So those are our A antigens. So if we have a scenario in which someone who is type A somehow encounters between their A antigens and anti-A antibodies, if antibodies come in and start recognizing these antigens, that is going to produce a big problem for us. So when this happens, when antibodies attack and bind to the antigens on a red blood cell, this is going to cause all of the red blood cells that are being recognized to start to clump together like this. So this is a process called hemagglutination. So this is not the same thing as blood clotting, so please try to not confuse the term. Hemagglutination refers to this clumping of red blood cells in an affected area once those antibodies have recognized their antigen counterparts, and this is a very bad thing. These red blood cells clump together. They can cause all sorts of problems within the circulation. They can clog up blood vessels and restrict blood flow. These red blood cells can be torn apart, releasing heme into the bloodstream, and that can get to the kidneys and cause all sorts of problems. So hemagglutination is something that needs to be avoided at all cost. So when you are considering, when do I give blood to this person or when should I not receive blood from this person, you do so keeping in mind that this is what you want to avoid. You want to donate and receive blood such that antibodies and antigens do not come into contact with each other. So let's consider two different scenarios here so that we can kind of get the idea here. So there are two very different types of blood types that we want to use as our examples here. So the first, I'll draw a red blood cell here. The first is type O negative. So if you are type O negative, you do not have A antigens, you do not have B antigens, and you do not have Rh antigens. But what you are going to have is anti-A, anti-B, and anti-Rh. So you have none of the antigens, but all of the antibodies. So if you are type O negative, and you've probably heard this before, you are what is called the universal donor. So the way this works is that when blood is donated, you are actually only donating the antigens. You are only donating red blood cells. Those antibodies get spun out of the blood plasma so that they are not being donated. So you really, if this is your donor down here, you really do not need to worry about those antibodies being transferred over into the recipient. You are only transferring these red blood cells. So the reason why someone who is O negative is considered a universal donor is because they are delivering and donating no antigens. So if you are delivering and donating no antigens, there is no possibility of those antigens which do not exist interfering or interacting with any antibodies in the patient. So someone who is O negative can donate their blood to any of the other blood types. You can donate to O positive, you can donate to AB positive, AB negative, you can donate to any of them. Since there are no antigens here, there is nothing to get in the way to cause hemagglutination. Now let's look at a second contrasting example here. So here we've got someone who is, I'm going to draw the A antigens. I'm going to draw the B antigens. And I'm going to draw the RH factor. So for someone who has all three antigens, this is going to make them AB positive. So someone who is AB positive is what we call the universal recipient. So to consider why this is, they've got all three of the antigens. They've got A antigens, B antigens, and RH antigens, but they're not going to have any of the antibodies. So because someone who is type AB positive, they have no antibodies against any of these antigens in their blood plasma, that means they can receive blood from any of the other 7 blood types, including someone who is also AB positive. So you can receive blood from someone who is type O positive, you can receive blood from someone who's AB negative, B positive, B negative, any of the other 7 and including the 8th blood type, you can receive it from all of them. Because since you have no antibodies in your blood plasma, at least none against these antigens, you can receive any of the antigens from any of the other blood types. But let's flip this thing on our head. Someone who is type O negative, let's say this is our recipient instead of the donor. Well, if you're type O negative and you are receiving a blood transfusion, there is only one blood type that you can receive transfusions from, and that is from someone who is also O negative. The thing to consider there is the fact that you've got all 3 antibodies in your blood plasma, so you cannot receive blood from someone who is type A negative, B negative, AB negative, O positive. You can only receive blood from someone who is O negative as well. Because even if you receive blood from someone who is O positive, that person up here has the RH factor, and that RH factor that they donate is going to interact with the anti-RH antibody, and that is no good. That is going to produce hemagglutination. On the other hand, if you're looking at someone who is AB positive as the donor, they can only donate their blood to someone who is also AB positive. So if you try to donate all of these antigens to someone who is, say, B negative, you are going to donate A antigens into a person that has the anti-A antibody, or you'll donate that RH antigen into someone that has the anti-RH antibody. So O negative is the universal donor, but the downside there is that they can only receive blood from someone who is also O negative. Someone who is AB positive can receive blood from any blood type, but the downside there is they can only donate blood to someone who is also AB positive. So this should cover our brief review on blood typing. So for the different types of blood types that we did not discuss here, I recommend that you make sure that you understand what types of antigens does that blood type have, what types of antibodies does that blood type have, and what are the different possibilities for blood transfusions in terms of what can you donate and what can you receive. So let's go ahead and sign off now, and I will see you next time.